Hello, Gen Bio. This week we are looking at an introduction to microscopy in lab, which is one of my favorite topics to cover in lab because I just think it's so cool. We get to observe things that would ordinarily not be able to be seen with the naked eye. So I really think this is a cool segment. So before we delve in, we are going to introduce you to the anatomy of the microscope. Okay, so starting at the base here, that's the bottom of the microscope. The microscope has what's called an arm, which continues up sort of the back of it. And then that will continue into the eyepiece, which contains ocular lenses. So inside the eyepiece, there's a lens which increases the magnification by 10 times whatever you have the objective set to. So if you look down here, there's what's called a revolving nose piece. And that nose piece has usually 3 or 4 different objective lenses that you can set it to. So the smallest could be 4 times or 10 times up to 100 times that you can set it to on these objectives here. So you want to be able to turn that to the appropriate objective lens to view whatever it is you place on the stage, which is right here. Okay, and remember that whatever it says here is going to be multiplied by 10 because you've got a 10 times magnification right there in the eyepiece. Okay. All right, so we often start with the lowest magnification. I'll just mention just because then you can see the whole picture, and as you need more detail, you can increase the magnification to the point where you can see everything clearly that you would like to visualize. Okay, so the stage, of course, is where you put your slide. There are little clips here that hold the slide in place. Okay, and in order to focus, after setting your objective, you will use the coarse and the fine adjustment knobs. So use the coarse one first, and then the fine focus in order to bring all the details into view clearly. Okay, I'll mention also that when you put your slide on the stage, if you were to adjust the coarse adjustment knob down so that it is pressing against your slide, you can actually crack the slide if you're too rough. So what I always recommend to students is that when they're first bringing the stage and the objective lenses together, so when you're using that coarse adjustment knob to bring the objective lens, draw that and the stage close together, that you don't look through the eyepiece. You actually put your head to the side and look at what you're doing to bring it down, bring them together first. And then when you need to look through the eyepiece, you can draw the stage away from the objective lens by turning the adjustment knob the opposite direction. Okay, and this avoids any potential cracking of our slides, which are not cheap. So that's how you bring your specimen into focus. I'll also mention that below the stage are the condenser and the iris diaphragm, as well as a light source. Okay, and down here is the light adjustment knob. So you can control the amount of light going in. The condenser is what focuses the light on your specimen, on your slide. And the iris diaphragm controls the amount of light that's actually passing through the specimen then. Okay, so there are lots of ways to properly light and properly adjust the focus of your specimen. So these are the basic terms that you should be familiar with for the microscope. And we're going to, in lab today, we're just going to first use a slide that will have a letter on it, just like a letter A or a letter E, to allow you to familiarize yourself with the microscope, get used to using the knobs and dials and lighting it properly. And so that'll be our first exercise, just to familiarize you with the microscope. Okay. Now, the next goal is to measure something that you would be placing on your stage. So you might have a slide that you prepared and you have a cell that you want to know how big that cell is. Well, there's a way to go about this that I will describe, and we will have you do in lab this week as well. So in the eyepiece, in the ocular, there is a specialized lens which has equidistant lines on it. Okay, so that's shown here, equidistant lines. So if you are looking through the eyepiece, you can have these equidistant lines overlay whatever slide you place on the stage. So here's your cell that you place on your stage, and you can view it along with the ocular micrometer because the lines are in the eyepiece and you have your slide on the stage. Now, these lines, though they are equidistant, you cannot conclude that they represent a specific number of millimeters or micrometers, because that will change as you alter the magnification of your cell. Okay, so what you need to do then is to calibrate the ocular micrometer to each of the different objectives that you might use. Okay, so if you have your microscope set to the 10x objective, what you then can do is place a stage micrometer on the stage. So you would have to remove the slide that has your cell, and then place a stage micrometer on the stage. That'll be on a different slide. Okay, so then what you would do, this stage micrometer has actual measurements, usually in millimeters. So this is, for instance, 1 millimeter, 2 millimeters, and you would place that slide on the stage and line it up directly with 2 lines on your ocular micrometer. So for instance, the 0 on the stage micrometer is lined up directly with this line on the eyepiece, on the ocular micrometer. And then you would have a point, it doesn't really matter where you have another line on your stage micrometer line up directly with a line in the ocular micrometer. So remember, this is what we're viewing through the eyepiece. This is what we're viewing through the stage, and you have to line them up directly. So that then what you can do is to count the number of lines on your ocular micrometer that equal a particular measurement on the stage micrometer. So for this example, I have lined up the 2 millimeter mark with, I say here, 42 ocular divisions. So hopefully I counted correctly. But the 2 is going to line up directly with 42 ocular divisions, right? So basically all you do then is you place the 2 millimeters above 42 ocular divisions, and then you can multiply by 1,000 to convert to the number of micrometers or micrometers that equal 1 division on your ocular. Okay, so you put 2 millimeters on the stage is equal to 42 ocular divisions, and this is just converting from millimeters to micrometers. Okay, so I got that each division in my ocular micrometer is equal to 47.6 micrometers in actual measurements. Okay, now this has to be at a particular objective lens. So this could be at the 10x magnification. You would have to redo that at the next greatest magnification, say the 40x magnification, because when you place your cell at a different objective, it's going to look like it takes up more lines on the eyepiece. Okay, so when you have calibrated each of your objectives to the ocular micrometer, then you can place your cell back on the stage. You can take off the stage micrometer, place whatever your specimen is, whatever you want to look at, back on the stage. And now we can say, okay, well, my cell takes up 5, 10, 15, 20, 25, say 25 ocular divisions, and multiply that by the number of micrometers per division to find out how big that cell is. Okay, and always remember you have to redo this process for each magnification. Okay, so then we're able to accurately measure anything that we place on our stage. Okay. All right, so let's move to our next slide. There are 4 different organisms that we're going to have you take a look at under the microscope today, or this week in lab. So the first one is Elodea, which is a common water weed. And the neat thing about Elodea is that if you take a leaf from the very tip of this plant, you can see very active living cells where they've got things moving around. You got chloroplasts that are actually moving around the cell, and you can visualize the cell actively moving and doing what cells do. So that's a really cool thing to be able to look at. And there are some anatomical terms for the Elodea cell that we would like you to be familiar with. The first is the cytoplasm, which is going to take up most of the clear internal space within the cell. And it's going to be held in the cell by the cell membrane, which you can't visualize too well. However, you can infer, for starters, that it is pressed up against the cell wall. So this is our cell wall in the actual microscope slide. Here it is, of course, in the diagram. And the cell membrane is pushed up against that to begin with, assuming that our cells are active and have a lot of water in them and not suffering from water loss. Okay, so cytoplasm, cell membrane, cell wall, and you will also look for the nucleus. The nucleus in diagrams is often shown to be kind of in the center of the cell. However, we are going to look for it as a grayish mass pressed up in the corner of the cells from the Elodea. Okay, so it's like a grayish mass pushed into the corner. You should hopefully be able to make out several of those nuclei. Okay, and then the vacuole, the central vacuole in Elodea, is a storage system for water and waste products from the cell. It's generally thought to be responsible for the turgor pressure. Remember, the water that is contained within the cell is allowing the cell to have a pressure up against that cell wall. We refer to as turgor pressure. So you will infer this region based on the fact that there are no chloroplasts present. So in the large grayish area, relatively clear structure, that is your central vacuole. Okay. Anything around that then that does not look like a nucleus, those are going to be lots of small chloroplasts, which you can actually observe traveling around the outsides of our Elodea cells. Okay, they're of course green, which make them readily observable and apart from the vacuole. Okay, all right, so let's move on to our next slide. One of the things we're going to have you do in lab this week is apply salt water to the outside of the Elodea cells. So if you remember back a couple weeks ago, you have to recall the concepts of osmosis and tonicity. Okay, and remember that one of the functions of our cell membrane is to control the movement of water and solutes into and out of our cell. So if you remember the concept of tonicity, I hope you all are thinking about what might happen if you add salt water outside of our cells. If you add salt, you're going to increase the solute concentration outside the cell. And so what would naturally happen is that water from inside the cell would be moving out to the external environment. Okay, so it's going to be drawn out of the cell. And in a plant cell, this results in the cell membrane pulling away from the cell wall. So now, whereas before in the Elodea, you couldn't really distinguish the cell membrane from the cell wall, you will hopefully be able to see that that cell membrane appears to shrink a little bit, shrivel up as the water exits the cell. So hopefully then you'll be able to see a faint line as shown here surrounding the internal contents of the cell, which has now pulled away from the cell wall due to the loss of water to the external environment. Okay, so we're going to hopefully be able to see that in lab today, this week. The second organism that we're going to take a look at is the Paramecium. The Paramecium is a one-celled, ciliated, so one cell, ciliated subtype of protists. So cilia are the little hairs on the outside. Of course, it's one cell. They live in freshwater environments. They feed on bacteria and other small cells present in that pond water. They can vary in length from 50 to 350-ish micrometers. And the cilia are what help them move through the water. They beat to move the Paramecium backwards through and forwards through the water. Okay. There are also a few additional structures which we would like you to look at in the Paramecium. Aside from the cilia, there are a macro and a micro nucleus, a contractile vacuole, usually two contractile vacuoles, an oral groove, and then you might see some food vacuoles that the Paramecium is using for nutrition. Okay. Then the contractile vacuole, I'm going to talk about again on the next slide, is a very neat structure that allows the Paramecium to exist in pond water. So let's move to our next slide. As I said, they live in a freshwater environment, which is obviously different from a saltwater environment. And what this means is that the environment is hypotonic, so less particles, less solutes, to the Paramecium. Okay, so water naturally, I hope y'all are raising your hands, water is going to go which direction? If it's in a hypotonic environment, less particles outside, water is going to be moving passively into our Paramecium. Okay, so again, this is not a plant cell. There's no cell wall. And so the potential hazard to this Paramecium is that if too much water enters that cell, this Paramecium could physically burst, right? Which would not be beneficial for the organism. So it has a structure that is responsible for allowing it to live in a hypotonic environment, and that is the contractile vacuole. And it often takes on this star-like appearance. You can see it here and here. The contractile vacuole is basically a water pump, and it pumps that water back out into the environment so that the Paramecium can exist without bursting in the pond water. Okay, so it's a neat feature of the Paramecium, quite the adaptation, and rather convenient for its survival in a hypotonic environment. Okay, so we're going to look at those this week in addition to the Elodea. The third organism that we will look at is the amoeba. The amoeba is another unicellular eukaryote. Okay, and these vary a lot in size, structure, and habitat. Okay, they can live anywhere from pond to seawater. They can vary in size from micrometers to centimeters, and their appearance will vary as well. We're going to study microscopic pond-dwelling amoeba. And they have a lot of the same structures that we looked at in the Paramecium, such as a contractile vacuole, a nucleus, food vacuoles inside that the amoeba uses for food, nutrition. But the neat feature for the amoeba that we would like you to be able to observe is the pseudopod, shown here. This would be one here, here. So these are basically extensions of the body that allow the amoeba to surround food, allows it to move around its environment as well as surround food to ingest. Okay, so here we have just shown the pseudopods have extended from the body, and they are in the process of surrounding this little food particle, which the amoeba is about to surround and digest by phagocytosis in order to absorb the contents for its nutrition. Okay, so the amoeba is a neat little organism that we're going to look at this week. It reproduces through binary fission, and hopefully you'll be able to see those under the microscope because they're pretty cool. All right, next and last organism that we're going to look at is the Volvox. It is a member of the chlorophyte green algae family. It also lives in freshwater ponds, and it forms these hollow balls because it's actually this is actually more like a colony of cells. So there can be up to 50,000 cells that make up this ball, and that they work together in a coordinated fashion to move and eat and reproduce. Okay, so this is a hollow ball, and inside there are daughter colonies, which then are offspring of the parent colony. So all of these little cells outside have flagella, which again work in a coordinated fashion to allow movement of the Volvox colony. So they're also a neat organism we're going to have you look at under the microscope in lab this week. Okay, so last slide to wrap things up, just summarizing what we're going to do. We are looking at the microscope and becoming familiar with all the parts that the microscope has, how to use it. We're going to learn to focus using a letter slide to start with, and calibrate the ocular micrometer to each of the different magnifications that are available on your microscope. Okay, and then we're going to start by looking at Paramecium, measuring those, and then we're also going to look at the other organisms, Amoeba, Elodea, Volvox, and a living Paramecium. The one we'll have you measure is already a prepared slide, so it will not be moving, but we'll have you look at that in a live specimen as well. And then we're going to apply salt water, as I mentioned, to the Elodea, and hopefully see what happens. All right, so that wraps up the presentation for this week's lab. If you have any questions, shoot me an email at cfry@lvc.edu. And have a great week.